All right, Dan, since we're recording, I'm going to go ahead and get started so that you guys can get forward. First of all, I want to thank you guys for taking the time. I know this has been a process that we began in July, believe it or not, and we've come a long way from where we began. For the audience that is out there, this is a webinar, so you'll only be seeing the four panelists right now as they move through their presentation. Do us a favor and down the bottom, if you have questions, type them in the Q&A box, and we will address as many as possible towards the end. As we mentioned, this is being recorded, so for anyone else out there who may not have been able to attend, we're going to, once the recording is downloaded, we'll send it out for parents to go ahead and take their time and review it. A couple of things that I just wanted to make very clear that with pool testing, this is an opt-in. I want to be really clear that no child or student will be swabbed, will be asked to be swabbed, will even be approached unless you have agreed to the opt-in form. Very clear on that. Second thing is, this is through the state funding program, the SASS program. So Hopkinton will be using no funds of our own to run pool testing. We had the benefit of essentially interviewing multiple vendors that the state approved, and each vendor had positives and negatives in regards to what they were doing and how they were doing it. We felt that this group of people in front of you tonight was the best opportunity for our district, least disruptive for our students, and a safe practice for everyone that was going to opt into this. So one thing I will ask both of this group and the community as we're moving forward is that a little bit patient as we implement, right? So it's a new process for us in our schools. It'll look and feel different than any other district. I know Concord began implementing it, but they're a much larger scale. So as we work through these, I'll call them bumps and bruises. I'll kind of compare it to running buses on the first day of school. Running buses on the first day of school, we're always late. We're always behind. We have new students riding it, people taking pictures, things of that nature. So as we roll this process out, obviously the first week will probably be slower. And then as we move through the weeks, we'll improve. So without further ado, I guess is the right way to say it, I'm going to welcome Maura, Dan, and Ben here to present. So thanks, guys.
Excellent. Thank you so much, Mike. It's great to be here with you all. My name is Dan Bram. I work with our team here at Ginkgo in New Hampshire, and I'm excited to talk to you tonight. And I'll kick it over to my colleagues to introduce themselves. Ben, you want to start?
Yeah, hi everyone. Ben Rome, part of the Concentric Ginkgo team, and oversee our K through 12 testing programs in the Northeast US, so in New Hampshire as well as Maine.
Excellent. And Maura?
And good evening, everyone. It's great to be here with you. I'm Maura Sullivan. I actually live here on the Seacoast in New Hampshire with my family. I am our Vice President of Operations at Concentric by Ginkgo, and so oversee operations of our testing business across the US and expanding internationally, and I'm one of the leads for our national testing program. Excited to be here with our New Hampshire team tonight, as obviously our state and our community is very near and dear to my heart, and we're just really looking forward to a really positive conversation and doing everything that we can to keep your kids safely in school. So thanks so much for coming out tonight and being with us, and thanks for having us.
Excellent. So I am going to share my screen now. There we go. And so what we'll do here is I'm going to walk through a presentation a little bit to talk about the program, who we are, how this will work, what it'll look like. I'll then show just a short video clip from how this actually is looking like in a school in Maine nearby, so you can get a sense of a little bit more color on it. And then we'll go to Q&A at the end and answer your questions. And so as we're going through this, if you have questions, feel free to put them in the Q&A section. And again, we'll get to those at the end of this presentation. So first of all, a little bit about us. So our company is called Concentric by Ginkgo. Our goal is to make testing accessible to every school in America every week. So we're working in a lot of states all across the country. We're now working in over 1,500 schools, which we're really excited about. And the thing I would just want to kind of point out here is that we're exclusively focused on K through 12 testing. So we generally don't take any other or clients. We're focused on what does this look like for educators? How do parents get involved in the process? How do we make this as not disruptive as possible so that ultimately your kids can spend more time in the classroom and less time out of the classroom or less time away from school. So a little bit about how our full program works, and then I'll get into the kind of nitty gritty of pooled testing. So first of all, pooled testing is the foundation of this. It's the idea with this is that we want to try to minimize the amount of time when your students are in school. If you decide to opt them into this program, we want to minimize the amount of time that they're spent out of the classroom doing COVID testing, obviously. So the way we do this is that we can actually group between 5 and 25 students in a pool at a time. And this allows us to get through classrooms faster and also just increases the overall logistical efficiency. We'll also then work with your district to provide rapid antigen testing. So I'll explain a little bit about how the pool process works. But in pool testing, it'll generate one result per group. We'll also provide rapid antigen follow-up testing. So how this will work is the idea is that the pool testing will ensure that we're getting through as many students as quickly as possible, as efficiently as possible. If any of the pools come back positive, we'll then work with you to we'll work with the schools to actually do rapid tests and quickly identify who in that pool is positive. And I'll show you a little bit about what that looks like in a moment. We'll also be sending nursing staff support to your schools to help with testing. So I'm sure you're all aware, I think everybody is overburdened these days and we want to help out as much as we can. So we will send nursing staff to assist in doing this pool testing. And then we'll also handle a lot of the public health reporting to make sure that your school's administrative staff can spend more time interacting with the schools and your students and less time doing public health reporting. So to give some more color on what pool testing is, so up to 25 students and staff are allowed to participate as well. We'll self-swab and they'll put all of their swabs in one single tube. So if you imagine all of these, kind of 25 students, up to 25, they will each swab their nose in each nostril. It's just a lower nasal swab. So we typically say if your child can pick their nose, they'll be fine with this test. And that one tube then will get run and we'll get one either positive or negative result for that tube. So from there, your school will get notified of if each pool is positive or negative. If that pool is positive, then when it's follow-up testing day, that rapid test I was just talking about, the nurses will then take those rapid tests and use that with the students who are in the positive pool to identify who exactly in that pool was the positive case. The reason we like this is, there's a couple of reasons. One is it protects privacy of students. But also it makes that, we don't have to then rapid test everybody all the time. And we only have to do that for students who are potentially in that positive pool. So here's just a little graphic too of what this will actually look like. So if you imagine all these swabs in here, so each student would take one of these swabs, they'll go nose swab, lower nose swab, and then put in that tube, and then that's the actual tube that would go to the lab. So it'll generate one result for that tube. So I think there's obviously a lot of different kinds of testing out there. I think the thing I would just want to, kind of highlight with this approach with your kids is that this is, first of all, it's free of charge to the school. It's pretty easy. We can typically get through a classroom in less than 15 minutes. So we really want to try to not disrupt your kids' learning as much as we possibly can. And then you all also will know that they've had a recent negative COVID test. So, I know my family, my nieces and nephews are planning on traveling for Thanksgiving. And kind of before and after that too, it can kind of be daunting to get those tests and have that peace of mind. So you'll also have that provided for them through the school. And on the rapid piece specifically, so again, pool testing, up to 25 students in a pool. If there's a positive pool, and only if there's a positive pool, we would then use rapid antigen tests on those students. In that test, you might have heard of the Abbott Buy Next Now test. We have some other rapid antigen tests that work the exact same way, just different brands that we can provide as well. But it's a very, very similar process where it's pretty much just the same lower nasal swab. You get a result back in a couple minutes there. So with that, I'm just going to pause here for questions, and also Ben, Maura, anything that you all would like to add there?
No, I think that you covered it really well. I think, the one piece that I would just kind of reiterate, and as Dan mentioned, the reason that we really like pooled testing is because it allows the district and schools to test students on a regular basis, right? So testing each week without a huge disruption to in-class learning, just given how quick the process is collecting all the individual samples into one tube. If you're, we've seen in other parts of the country and other programs, schools and districts rolling out, a regular rapid antigen testing program where you're actually using the rapid antigen tests every single week. The tough part about that one is it takes a lot more staffing, like human resources, like a lot more nurse support to staff it up, which can be tricky, and also there's just a constraint on the supply of those tests. And then lastly, it just takes a lot longer, so it's going to be more disruptive to the in-classroom learning. So what we've found, and we've been doing this since last fall, and kind of in a number of different, as Dan mentioned, over 1,500 schools. What we've found is the pooled testing it to be a great balance between being able to test regularly, on a weekly basis, while minimizing, disruption and burden to the school.
I think the only thing I'd add to that is just to emphasize again, like quick amount of time. Like this is supposed to be really hardly disruptive at all to the regular classroom and learning environment. And when we designed this over a year ago, that was a big factor that we took into consideration, as well as the cost structure. So when we looked at this, it was before there was federal funding in place, and we said, we've got to make this affordable for districts across the country. At the time, an individual PCR test was running north of $125, and you could do a pooled test for roughly what comes out to about $6 a kid, if you assume about 25 children in a classroom. And so that was something that was really important to us, is to let's make this sustainable and affordable for districts. But to be clear, since that time when we designed it, there has been federal funding that has been applied to school testing across the country, which New Hampshire got a portion of. And so none of this comes at any cost from the actual district. It is 100% federal funds.
Excellent.
Dan, I'm going to jump in and answer a couple of these because they're mine, more district specific questions in regards to, I'll call it supporting younger students. And the question is, it will really be as simple. We did send out a couple of videos that showed more specifically what will happen within the classrooms. But for parents with younger students, that's where I'll be working with our staff internally, both the nurses and or classroom teacher at that time, to help support students who have opted in to feel comfortable with the testing procedures, walking them through it, supporting them same way we would when we're introducing something new, to make sure that they feel comfortable with the process. So that will be taken care of on our internal processes. And I actually have a question, Dan, now that we've been talking about it. Is it the same staff that come back each week per school, or is it scheduled with whoever's available?
Yeah, we try to make it the same staff who comes back every week.
Great.
Mike, can I take the question from Elizabeth here in the chat, and then maybe Ben, maybe you could take the one from the anonymous attendee in that order. Does that sound good?
More, I just kind of addressed Elizabeth's. So if you want to add to it, sure.
Yeah, the only thing
Okay.
I was going to add, Elizabeth, is so it's a great question. And we'll make sure that we send, I think, Mike, you said you had the videos, but we'll make sure you have a copy of them just in case. But we've had a number of kids actually in the sort of 5-6-year-old range that have said, it's like digging for boogers. And we've utilized some of the videos actually in the classroom at times to kind of introduce it to kids, or someone will demo that. But we've actually seen in it always makes me smile, like thinking about these. We've actually seen in a lot of classrooms across the country where kids kind of get really into it. They feel empowered, but it's the comment is it's like getting boogers. So yes, I think the short answer to your question is yes, it is pretty straightforward.
Excellent. And to probably give a little bit more color to this. So sorry, apologies for the ad that started playing during the presentation, but I have that video back. So I'm just going to play this real quick. Just so folks have that. Let's see.
It is already underway in some of Maine's schools weeks before classes begin.
Pooled PCR testing is designed to detect the virus before students are contagious. News Center Maine's Carly Dion gives us a look at how this program works.
Any kid who can pick their nose, which is most, can do the swab, and it's really easy.
Courtney Arcaro is the school nurse at Setamocha Middle School. She's in charge of conducting weekly pooled PCR tests for its summer school program. Every Tuesday, it's testing day.
All the students who have parent consent swab their nose, put their swab into the tube, which is not labeled with any student identifiers, and then those tubes get sent off.
Results come back within 2 days, and in just that time, it can detect the virus before kids infect others.
So if you get a positive pool, you then go in and rapid test the students that participated in that pool, trying to find the positive case. And then say you find the positive, that student does have to quarantine.
Maine Department of Education school nurse consultant Emily Poland says the state can provide extra support if a school needs it.
That can be administered.
So that's I just wanted, the rest of it's like Maine specific, but wanted to make sure that you'll have the kind of that context of what that actually looks like in terms of students taking the test there.
Ben, can you take the DNA question?
Yeah, absolutely. So there's a good question here about, your tests rely on DNA from a swab, what happens to that swab and the associated data, and do you link the student to the material on the swab that's gathered? And, that's a great question, and that's actually one of the reasons that we really like pooled testing. So the way that it actually works is, this is the pooled testing tube. Dan showed a picture of it before. All of the individuals in a classroom, all of their swabs go into that tube, and that tube just gets a name, right? So that tube is called, pool A, pool B, pool C, or Mrs. Smith's class, or whatever the school wants to name it. We actually don't. So when the lab runs the test, they scan the barcode, and the result is associated, with that tube serial number and that the name of that tube. We actually intentionally don't collect any of the information as to which individuals are in that pool. And so really what happens is, on the school side, when they're doing the testing, that pool A, or Mrs. Smith's class, were these individuals on this date. And that's something that the school keeps on their end, so that if there's a positive pool that comes back, they can identify, which individuals need the follow-up testing. But again, we're not able to see, nor would we want to, which individuals are actually in that pool. The other thing, and it's like a bit more of a technical point, but basically when the lab is reading the test, you're really just reading the viral RNA, so you're not actually analyzing any human genetic material. But it is kind of a moot point, as again we don't know, which individuals are in that tube.
So there's a couple more probably from me rather than you guys. In regards to question was, will we be working with athletic teams? That's a topic of discussion currently at the school board level. We are examining practices and procedures on whether that is a discussion. They will not specifically work with an athletic team. It would be more about an opting in of a student. So, but that discussion is going to be held at the next school board meeting. I think one of you guys should answer Anna's question, if you don't
Yeah,
mind.
so why don't I'll kind of take Anna and Nicole's, and then Ben, do you want to just kind of add on to the back of that? Because I think you sort of touched on this earlier. So Anna, one of the reasons that we don't initially do the rapid antigen, one of them is a supply chain issue. It's simply there's a shortage of tests both across the state and across the country. And so to do rapid antigens at the scale that would sort of be required is just not something that is sustainable. Ben also, I think, touched on this earlier. It actually takes more time to do, and is I don't know if anyone's ever done a Buy Next Now. I just did one like 3 days ago. It's a lot more sort of labor intensive, and it's not as simple, kind of going back to Elizabeth's earlier question, to just have a 5-year-old do a quick self swab. It would require kind of significantly more sort of supervision to make sure you're pouring the component into the test kit the right way, to actually make sure that the results are accurate. We use them as sort of the right after the pool testing, because that's a much smaller group to then test. So rather than testing all 250 kids at an elementary school, so to speak, and with 250 rapid antigen tests, we can do pooled testing in 10 classrooms of 25, and then let's say of those 10, 9 have a negative pool and 1 is a positive, and then the school can then go and make sure that 1 is tested. And then Nicole, the question is, if a positive pool is found in a classroom, how quickly will you return to test that pool? So the lab we'll get the answers back, or I'm sorry, the results back to the school within no more than 48 hours. So it's a guaranteed 24 hours to get to the lab, and within 24 hours to get it back. It's often less time than that. But that is the kind of guarantee that we give as to when the school will get the results. And again, just to reiterate something that Ben said, the only thing that we see on our end is just, pool A, or, Mr. Robertson's 4th grade class. We don't know who's in Mr. Robertson's 4th grade class. All of that information, the school maintains, and we simply inform the school, hey, of your 10 classrooms that were tested, 9 are negative, and pool E, which is Mr. Robertson's 4th grade class, is positive. So let me pause there, and Ben, just anything to add to, what I said about the rapid antigens, and then maybe explain a little bit more about the testing that we would do when they get the results.
Yeah, absolutely. So yeah, the only things I would add, so on the second piece, in terms of results returned and when we'll return to do testing. So the school will, say a school's testing Monday, a school will almost always get the result back sometime on Tuesday. So the way it works is we have a courier service that picks up the test on the day, on the pooled test on the day of testing, and transports that so that it arrives at the lab on the same day. So it'll arrive at the lab, Monday afternoon or evening, and then the school will get the results on Tuesday. Because the results may come in on Tuesday, sometimes before kids are out of school or after kids are out of school, we send staff on the following morning. So if it's a Monday test day, the Wednesday morning to do any necessary follow-up tests. What we can also do, and we work with districts on this, is if we're consistently seeing that results are coming back, say Tuesday morning for a Monday test, that's when we can make the adjustment to have staff coming in, on Tuesday to conduct the follow-up test. So we have kind of some flexibility on that front. And then on the rapid antigen and kind of why we don't just use that test right off the bat. I think Maura summed it up well. The one other piece I would add is, there's been a ton of different research on the accuracy of the rapid test, right? And so one of the things, and you can read all the different studies. One of the things that some studies have seen is that if you're using a rapid test on a broad scale asymptomatic population, their accuracy can be, it isn't as good, right, as a PCR test, of course. When you're using a rapid test on a smaller population, say a pool of 10 or 15 or 25 individuals, where you have pretty good reason to believe that there's a positive in that group because the pooled PCR test identified it, your predictive value of that rapid test goes up quite a bit. So I think it's all those things that that kind of have led us to, over the last year, kind of hone in on this model of program.
We've had a couple questions come in regards to students being quarantined until the individual testing can be done. Ben kind of hit that on the head by saying, once the results are in, typically on that Tuesday, there would be someone there in the morning for, I think we were using Mr. Robinson's class to do the rapid testing. So at that point, we would be able to test any parent or any students that have opted in, and or I believe, Ben, correct me if I'm wrong, that evening that class would be notified and any parent could opt in that next day if they weren't already in to receive a rapid.
Yeah, that's right. So, after a positive pool comes about, a parent, could opt in. You can opt into the program at any time. So a parent could opt in so that, their child could still receive the rapid follow-up test the following day.
And those are just still some of the internal practices that I spoke about at the opening, as we try and figure out how, I'll say we have the foundation in place, but is it what's best for Hopkinton in the schools? We can figure that out, meaning if that classroom is positive, do we bring them in, obviously not on the bus, through parent drop off, things along that nature. We're still in process of finalizing those plans so that it works well with Ginkgo, which we've selected. So. Sorry, we're just taking a second to read some of these. Just hang on. That's a great question. And I guess both of them kind of fall in the same. With the other districts that you've begun to use, is there a prime number of percentage of people that you've seen with better effectiveness than others?
Yeah, I can take that. That's a great question. So there's no hard numbers. There's no threshold, right? That, above this percentage, pool testing is effective and below it, it's not. So the short answer is, intuitively, the more folks that are participating, the better. What I will say is a couple things. Even at a lower participation rate, 10, 15, 20%, you're still getting a good snapshot of what the positivity level in the community is. So ideally, you're testing many more people than that. But what that does do is help you see, you know, over time what the rate of COVID is in the school, and then help the district and the school make decisions based off that, you know, in terms of some of the other safety measures that, you know, they may want to put in place or remove or what have you. So I think you know, the low levels of participation still valuable, still effective. The one other thing I'll just mention, often what we see with schools and school districts is, you know, first week or two, participation rate maybe on the lower side, and then after, you know, the community and teachers and students kind of get to see the process in that first week or two, we start to see the consent rates and the participation rates rise quite a bit.
And there's a couple more data questions that, you know, Ben did a good job answering there, but in regards to our data, which I've said repeatedly at board meetings, you know, we have our dashboard that you see, and you've seen our cases have remained significantly low. Through pool testing, I will regularly update the board on number of opt-ins, percentage of classrooms in which they've had a positive or not. So that data will be coming out through the board meetings and the updates.
Yeah, I can take the question. There was one other question just about the kind of any data behind like the efficacy of pool testing that we've seen. So I'm happy to also just add on a little bit on that. So we haven't, at this point, like we haven't done any, like there's not any published studies that we've put out about, you know, like peer reviewed about the data on that. What I can say is a few different things. So anecdotally, you know, what we've seen and heard. So we've seen with a number of districts that we work with, positivity rates drop, you know, starting when they started implementing pool testing, you know, seeing pooled positivity rates of, you know, 6-7%, and then over the next few weeks dropping and steadying around 2 or 3%. Again, there's a lot of different factors, right, that could play into that. So it's not it's more of a correlation, right? It's not an official study, but anecdotally we have seen that. What we've also seen and heard from other places is that it does reduce days missed overall by students, because it allows you to pick up asymptomatic testing in general, right? Allows you to pick up infections before you might otherwise would have, and help prevent the spread there.
Okay. On the last question here, it's going to be me and not you guys again, in regards to return to school. So whether they're asymptomatic or symptomatic and they test positive, the positive is a positive result. So the positive result would then follow the same practices and procedures we have in place through the district in regards to the amount of time you would need to quarantine per DHHS. The kids that will also be in that class when they take the rapid, if they're negative, then they're negative. So that would be a monitoring. We would do a close contact evaluation, and we would tell your students to evaluate. We would ask the parents to evaluate their students' symptoms over a certain period of time. That would be the same practices that all 3 nurses have in place. So really, this would only be identifying asymptomatic students and then following our internal practices already. Okay, so I want to thank the group for taking the time. I'll reiterate it for anyone that may have joined late. We have recorded this. Once we process it, we'll be sending it out through our communications to the community. And from this point tomorrow as well, either tomorrow or Thursday, tomorrow, you'll be getting the opt-in form through communications from the district. So those are the next steps, and then we look forward to beginning this partnership with Ginkgo. If you have any more questions as we dismiss here, please feel free to send them to the school nurses, and then they will work with me in regards to answering them as best we can. We can bring Dan, Maura, and Ben back in to further any information if needed. But I just want to thank you guys again for taking time out of your Tuesday evening to help join us in this district. And I look forward to working with you.
Well, thanks, Mike. And I know just on behalf of our team at Ginkgo, it's a real privilege for us to get to serve your community in Hopkinton. So thank you for inviting us here this evening. Thank you for bringing us to your community and into your schools. And this was a program that was very, very proudly designed, by mostly parents for other parents. And so, when we built this, it was, how would we think about this in our own children's schools? And so we're very grateful for the opportunity to work with you and partner with you. And we're happy to come back, Mike, as you said, if that's at all helpful, but really just appreciate you having us here and allowing us to serve you and your families.
All right. On that note, have a great Tuesday night, and we'll be in touch tomorrow. Thanks, guys.
Thank you.
All right.